And I'll just give it a moment to let YouTube catch up, and then I'll start. Okay, everyone, good morning from the United States. Good afternoon, good evening in other parts of the world. Thank you for being with us today. And as a reminder, could get some feedback. So I'm going to ask people to please make sure that you mute yourself. Good morning. This is the FIND seminar, and my name is Lauren Hayes. I'll be the host for today's seminar. And we're really happy that everyone's back after a bit of a break. This is the third seminar of the FIND series. And before I announce today's speaker, I just have a few announcements about the seminar. And I'll go over some of the seminar functions since we have a lot of new faces joining us this time. So first of all, you know, the FIND has grown. It's now up to about 500 registered. We have 500 participants registered on our email list. And on the last FIND, you know, we're averaging, you know, upwards of 100 people a day. So this is really exciting. The FIND structure is we have speakers each week who give a talk that's about 45 to 50 minutes. And that's followed by upwards of 60 minutes of discussion. We live stream this by YouTube, so hopefully that is going through now. And of course, we're on Zoom as well. We have a really exciting series, we think, set up for this coming year, this time. I'm going to click a link to the speakers for the series this term. As I'll talk about in a moment, we have our first guest will be Raghavendra Gadagkar from India will be presenting. Next week, we have Shenfeng Shen from Taiwan will be giving a talk. And you can see that after that, we have, you know, the speakers. We then have Jennifer Smith from California in the United States giving a talk on comparative social evolution in mammals. This is followed by Juan Carlos Reda, I believe from Argentina, giving a talk on arms races and birds. Clint Pennock will be up following that, giving a talk on social ants. He's at Kennesaw University in Georgia. Allison Bell will follow by giving a talk on family life in sticklebacks. We then have Annalise Berry from Smith College, United States, talking about vole mechanisms or mechanism sociality in voles. And then we have Andy C. Giving a talk on individual differences in behavior and social dynamics. The week after that, our aim is to have a mini symposium on long-term research studies. So we're working on the framework for that and the organization, but hopefully we'll have something further to tell you later in the term, maybe sometime in October. After that, we follow up with Julia Fisher, talking about baboon research. Monique Borgerhoff-Mulder will talk about polygyny and anthropology. And then I believe we wrap up this term with Wolfgang Goeman talking about physiological mechanisms and sex role reversals. Oh, sorry, we actually have 3 more I forgot about. After that, we have Hannah Coco from Switzerland giving talks about lifespan, selection for living shorter lives. Noah Pinter-Walmer will then talk about spatial and ecological features, animal movements. And then we wrap up with Valeria Romano talking about social networks and hunter-gatherers. So we have, I think, a really remarkable series of talks coming up. I'm looking forward to them. All of this, as you can see, is found on our webpage, which I'm showing you today. We encourage you to go check out this webpage. It has lots of interesting information, useful information regarding the seminar. We have our list of past speakers from the fall 2020 and spring 2021. And then if you click the link that says FIND Teaching, if you're interested in using FIND tools in your classes, we have links to various resources that may be useful to you. We have a certificate for students that want to register and get credit for this. All the readings that are provided by the speakers are posted here. So if you click on this link, you'll find the reading list for the fall 21, spring 22, and fall 2020. And you click on those and you can get access to those references. Other resources are, we have teaching slides prepared by some of our speakers that can be used in your classrooms. We have a resource that I used in one of my classes where I talk about I give you a sort of an instruction guide for how to use FIND in an undergraduate behavioral ecology class. This is how I did it in the spring. And I posted here so that folks could get some ideas for how they might incorporate this into their classes. So there's a lot of resources at the website that are available. And I'll link one more just to give you some news. We had a paper come out about in ethology this past summer. So in July, I think it was accepted. Some of the FIND folks got into a working group and we produced a paper on the use of remote seminars and classes. So some ideas for how you can use this information in your classes. Hopefully it's highly accessible to everyone and can help us to, be more creative in how we teach animal behavior. And I want to thank Facundo Fernandez Duque for his hard work in producing this website. He did all the work on the website, with input from the hosts, Eduardo, Carson, and myself, and has done a great job with this. Couple of other quick announcements. We have I have one other piece of exciting news. As you know, those who've been around us know that Carson Schraden, Eduardo Fernandez Duque, and I were behind the start of all this. And we realized quickly that the amount of work going into this was pretty intense. There was a lot of it. So we sought out a fourth co-host. And starting in the spring, we're thrilled to have Adriana Maldonado Chaparro help us with the FIND. Adriana is a professor at the Universidad del Rosario in Colombia, and also an associate of the Max Planck Institute of Animal Behavior. Adriana is going to bring her expertise in social structure to the game, and she has a wide background in research on mammals and birds. So really excited to have her on board. And Adriana, if you're out there, do you mind just saying hello, since you'll be putting you on the spot today, Adriana?
Hi, Lauren. Thanks. So as Lauren said, I'm at Universidad de Rosario in Colombia, and I'm really, really happy to be part of this awesome group that put together the FIND seminar. So if you have any questions or just want to, get in touch, feel free to do it. Thanks for the invitation as well to be part of this.
Yeah, thank you, Adriana. We're really pleased that you're joining us, and I think you'll see it opens up, it opens up the scope of the kinds of speakers we can bring in. Hopefully, bring in new ideas. So we're really excited about having her on board. All right, so that's enough about the FIND. So I'm going to introduce today's speaker. We are thrilled to have Rag Gadagkar with us today. Rag is a professor at the Center of Ecological Sciences at the Indian Institute of Science in, I believe, Bangalore. He earned his PhD at the Indian Institute of Sciences and a master's and BS at Bangalore University. Those of you who don't know his work, you'll come to find today how impressive it is and how interesting and exciting and diverse it is. He studies the evolution of social life in insects, also dabbles in understanding insect biodiversity, and has even done some modeling of genetics and developmental biology. Rag has been amazingly productive. It was really fun to talk to him this morning about what he's done. He has 3 books, one of which I'll mention in just a moment, and over 300 publications in peer-reviewed journals that range from the behavior journals to Tree to Nature, Science, and Proceedings of the National Academy of Sciences. So highly prolific and successful scientist. He's had a number of awards and honors as part of and throughout his career. He's past president of the Indian National Science Academy. He's a fellow of the National Academy of Sciences in India. And interestingly, he's an international member of the US National Academy of Sciences, where he now is on the editorial board and even edits papers for, acts as an editor for papers for the journal PNAS. He's also a member of the American Academy of Arts and Sciences. Rag has also had an amazing impact on students throughout his career. He's supervised 25 graduate students over his career, and has in his lab, been quite productive. As I mentioned, he has a recent book that came out, and he'll be talking a little bit about that today. So I'll just introduce it briefly. It's called Experiments in Animal Behavior, Cutting Edge Research at Trifling Costs. Okay. Oops, I'm going to cut the website here. He also maintains a blog, which I found really interesting, called More Fun Than Fun. And I actually, when I was going back and learning about his work, I was reading some of these, and they're quite fascinating. He does twice a month, he writes these blogs for The Wire, and actually it delves into topics that integrate science and history and really reaches out to a broader community on how science works, okay, and makes connections. And I found that really fascinating. In fact, there was a really interesting read in here about his history of reading a textbook and how it influenced his career, and then learned about sort of the nefarious behavior of that scientist later in life. So it's really interesting to make connections with humanity in his writing. He's also the founder of the Center of Contemporary Studies, which at his institution, an amazing program that fosters interactions between the natural and human sciences. So he would bring speakers from all different disciplines to his institution to talk to the students there and learn not just about science, but also history. And the way he described it to me this morning was that, the speaker would present half the talk on their discipline and then half the talk on how they influence their discipline. And to me, that was really powerful because it represented a true liberal arts way of thinking. And that's something that I think we could all really benefit from, not just focusing on the science, but thinking about all elements of learning. And I'm going to stop sharing my screen and let Rag share his screen, because now I want to introduce the title. And the title of his seminar today will be Direct Fitness Options for Workers for Sterile Workers in the Primitively Eusocial Wasp, Ropalidia marginata. And with that, I'll turn the seminar over, and thank you all for being here. And Rag, thank you for giving us a talk today.
Thank you. Can you hear me okay? Wonderful.
Yes.
Thank you, Lauren, Eduardo, Carsten, for this invitation. I'm really happy to be here, to be part of this family, and I look forward to participating in the future lectures this semester. I could not do so last semester because every Tuesday I had something else, but now I'm going to reorganize that and be part of this family. So it's a real pleasure to speak to you about the works, some of the work that my students and I have been doing. I have called this Direct Fitness Options for Sterile Workers in the Primitively Eusocial Wasp, Ropalidia marginata. You will notice that there is a bit of an oxymoron in the title, which is deliberate. Direct fitness and sterile don't quite go together, and that's why the sterile is in quotes. And I'm going to really talk about this oxymoron today. But let me begin with some very simple introduction. I always like to begin with something that people already know and then connect to something new. So as you would all know, a fundamental feature of eusocial insects are the so-called truly social insects like ants, bees, wasps, termites, and quite a few additions in more recent times. A fundamental feature of these eusocial insects is what we call reproductive caste differentiation. So differentiation of members of a group into two kinds, the so-called fertile queens, which are reproductive, and the sterile workers who are non-reproductive. So this is the fundamental division. And this division raises, of course, the famous paradox of altruism, because these non-reproductive individuals are, in an evolutionary sense, in a genetic sense, acting altruistically, spending their lives working for the welfare of the colony, taking care of the offspring of the reproductives, but not having any kids of their own. So the fundamental question, which bothered Darwin and many people since, is how does natural selection favor the evolution of such altruistic workers? On the face of it, natural selection should get rid of them as quickly as possible, but that does not seem to happen. And the prevailing theory, which is a very successful theory, despite some minor controversies, the successful theory goes by many names. It's sometimes called Hamilton's rule, sometimes it's called inclusive fitness theory, and sometimes called kin selection theory. It's really very similar with minor variations. So I'm going to use these three terms interchangeably for today, but let me explain this very briefly using the first name, that is Hamilton's rule. So W.D. Hamilton, as all of you know, in 1964 came up with this simple, elegant mathematical inequality: RB minus C must be greater than 0 for natural selection to favor an altruistic trait, where B is the benefit of the altruism to the recipient, C the cost to the donor, and R is the genetic relatedness between donor and recipient. So if B is devalued by R, if the benefit is devalued by the relatedness, then that should be equal to the cost, or it should be greater than the cost in order for altruism to be favored. The implication of this, of course, is that Hamilton was expanding the concept of inclusive fitness. He, in addition to the direct fitness gained through offspring production, he added an indirect component which can be gained through aiding genetic relatives. The direct component, of course, is the standard Darwinian fitness, and the indirect fitness is the new thing that Hamilton added. But the surprising thing of this was that you could get fitness without having any kids. You could get fitness simply by taking care of your relatives. Quite naturally, people became excited about the indirect component. The direct was old story. So all the attention was directed towards understanding how indirect fitness works, how indirect fitness is accumulated. And therefore, all the attention was on this. But there is a little bit of a problem in focusing all attention on this, because this process of reproductive caste differentiation comes in at least two varieties. In the so-called highly social insects, the differentiation of reproductive and non-reproductive queens and workers is pre-imaginal. That is, it is before the adult stage, in the larval stage, and therefore the process of caste differentiation is through developmental regulation, and workers seldom have any direct fitness options. Individuals become adults either as queens or workers, and they can't really exchange their roles anymore. But in the so-called primitively used social insects, caste differentiation is post-imaginal. That is actually happens in the adult stage, and therefore it is through social regulation rather than through developmental regulation. And for that reason, workers often have some option of getting at least some direct fitness, and therefore at least some attention should be paid to this. Nevertheless, the possibility of direct fitness has usually been ignored. There are, of course, exceptions, but usually it's been ignored, even in primitively eusocial species. We sort of look up to the highly eusocial species and model our research, our discussion, our questions all on the highly eusocial species, which have, of course, a much longer history of being studied. Now, my students and I study the so-called Indian paper wasp, which is a primitively eusocial wasp. And in this wasp, therefore, we have become interested in looking at the possibilities of direct fitness. And we think there are additional reasons why we should focus on direct fitness in this species. First of all, we find that in a colony, genetic relatedness can be quite low. Many years ago, we showed that queens can mate with 1, 2, or 3 different males, mix the sperm of these males, and produce different factory lines of daughters with the colony, so that the relatedness between the daughters who work for each other can be quite low. In fact, it can drop from the theoretically expected 0.75 because of haplodiploidy to almost 0.5, which is what you find in diploidy. So there is no special advantage of being haplodiploid when it comes to dispensing altruism in this species. Furthermore, and even more importantly, we found that not only are multiple fathers in a colony, there are also multiple mothers. And this is because being a primitively eusocial species, having post-imaginal caste differentiation, any worker can in principle become a queen. And indeed, there are many opportunities for workers to do so. From time to time, queens are killed or are driven away or die, and one of the workers becomes the next queen. And here, because of this, and because we mark all our wasps, we actually have constructed royal pedigrees of these species. I'm very fond of saying that this is the first royal pedigree of an invertebrate. And here you can see that we had queen number 1. She was superseded by her daughter. The queen number 2 was superseded by her sister. Queen 3 by another sister. But queen 4 by a niece. And queen 7 by her cousin. So all these things happen. And when this happens, the workers do not stop working. They continue to work for the offspring of the new queen as if nothing had happened. In fact, you cannot even detect this transition except for a day or two. You cannot detect any disturbance. Because of that, the relatedness within the colony drops further. In fact, we have been able to show purely by pedigree analysis that the relationship between queens and their immediate predecessor can that of daughters, nieces, sisters, nieces, and cousins. And therefore, the relationship between the workers and the brood they rear can be very diverse. Brood can be their sisters, brothers, nieces and nephews, cousins, cousins' offspring, mother's cousins' offspring, mother's cousins' offspring, and even mother's cousins' grand offspring. I'm very fond of saying that Ropalidia marginata will put to shame any Indian joint family because they live in this extremely diverse. And when you put all of this together, you find that the intra-colony relatedness really drops below 0.5, which means that there is all the more reason for workers to look out for possibilities of direct fitness. So indirect fitness, therefore, may be inadequate to drive social evolution when worker queen brood genetic relatedness is so low. Therefore, direct fitness is possibly and may be necessary for the evolution of social behavior in such species. We have therefore begun to vigorously focus on the possibility of direct fitness for workers in this species. In the species that we study, workers can get direct fitness in two different ways, in at least two different ways. First is new nest foundation. Workers can leave their nest of their birth, leave the nest in which they're working to start their own, either solitarily or in small groups of individuals. And in the solitary case, that individual will now get direct fitness. She was getting indirect fitness in the mother's nest. Or if there's a group of individuals, at least one of the individuals will now get direct fitness. The second way in which they can get direct fitness is through queen succession, which I already mentioned. Workers can overthrow or replace existing queens and become new queens in their own natal nest and transition from indirect fitness to direct fitness, even in the same nest. So either by going to and building a new nest or in the same nest. In principle, there is a third way to get direct fitness, which we see in Polistes and other species. That is for the workers to sneak in, covertly sneak in a few eggs, even though they have a fertile queen. We have never seen that happen in our species, in spite of thousands of hours of observations by dozens of students. So we think these are the two main ways, and these are routinely used by workers to get direct fitness. I would say to augment their indirect fitness with further direct fitness. So I want to talk about each one of them separately. So let me begin with the nest foundation option. This is very interesting for us because what I'm going to tell you now is actually very recent work. For this option of queen succession, we have been working for a very long time. And the reason why this is rather interesting. We work with these wasps wherever they are. And in order to build a new nest, they leave their nest and go somewhere. And we don't know where they go. We see them leave, but we never see where they go. We see new nests initiated, but we don't know where the wasp came from. So we were not able to make the connection. And in the lab, the problem is the cages we were using were too small to give room for two nests. So we did a very simple thing. We constructed a large cage, and then we have been able to solve this problem. We actually now study new nest foundation in three different contexts. First, we call it voluntary nest foundation. A wasp or a group of wasps on their own leave and start a new nest on their own. The second context is we ask, when wasps are having indirect fitness, do they lose or retain their potential for options of direct fitness in the future. So this is a separate kind of study. And in the third, we call it involuntary nest foundation, where we experimentally put together chosen groups of wasps, sometimes one, sometimes two, sometimes three. We put them together and allow them to build a nest and observe their behavior. So in these three different contexts, we have studied this. So again, I'll take them one by one. Voluntary nest foundation. It is voluntary nest foundation, which was very difficult to study in the wild. It was very difficult to study in these small cages because there apparently is not enough room for someone to live and for two different nests to coexist. All we did was we built a large nest where I could put the wasp nest and my student inside the cage, and observations are made inside. And now there is actually space for them to go and do that. And this changed everything. This is one of the first students who did a PhD on this subject. You can see behind her is the new kind of cage, and next to her is the small old kind of cage. In these so-called walk-in cages, we observed in the first study 29 new nest foundations. We had never seen even one before. Starting from 9 parent nests, 9 of them were single foundress nests, 20 of them were multiple foundress nests. Perhaps our most interesting result from this, let me say it upfront, is that our observation is that wasps from pre-foundation aggregations away from their original nests, where they feed and engage in dominance-subordinate interactions. So they leave their parent nest, they have this temporary place where they settle their dominance relationship, and then they decide to start a new nest. Some of them may opt out because they lose in this and go back to the old nest. So my student kept track of the events happening in these walk-in cages ever since we transplanted a naturally occurring large nest, which we call the parent nest. And so she kept a diary of what is happening. So for example, on each day, what happened? Parent nest transplanted on December 13th, then what happened on January 8th, new nest started. She keeps track of this. This is a simple example. The most complicated example is here, where lots of things happen every day. So constantly she keeps track of this every day. And by analyzing this, she documented several unusual behaviors. One, as I said, is wasps congregated outside their parent nest and showed dominant-subordinate interactions before initiating multiple foundress nests. Of course, if it was a single foundress nest, she just went and started it by herself. But if she had to take some workers with her, then there was need for this kind of pre-nest foundation conference. Solitary foundresses, of course, never participated in such off-nest aggregations. The most dominant wasps in the aggregations always became queens of the new multiple foundress nest. So that question was settled even before they decided to go. And solitary foundresses and future queens of multiple foundress nests engaged in self-feeding behavior outside the parent. Normally, wasps never feed outside. Even the foragers go bring food, come back to the nest and share it with others and feed there. But in these nest aggregations, there was this feeding. With this information, we were able to do other experiments. So the second context is retention. So what we did here was that we asked how long do wasps retain, or when do they lose their reproductive potential if they engage in indirect fitness? Do they lose it? We recorded the behavioral profiles of all wasps on 6 naturally occurring nests in Ropalidia marginata. And then we took away all the wasps, not the nest, all the wasps, and we isolated every wasp into a little individual plastic box. We know that individuals in these little plastic boxes will build new nests, lay eggs, and rear offspring. But these are for newly closed wasps. Now we took wasps from all ages, from the mature nest, and we gave them all an opportunity to initiate eggs and ask who does and who does not. Some of them did, some of them did not. And that gave us an opportunity to try and understand what prevented some from doing so, what allowed some from doing so. So when we did a statistical analysis of this, and we checked out various predictors of the ability to build a new nest in the plastic box, of the ability to retain your reproductive potential, such as age, their mated status, their dominance behavior, the amount of work they do, the amount of food they ate, and interactions between some of these variables, we found that there were only 2 variables which were significant. One was age and the other was feeding. Well-fed wasps, there is natural variation in feeding rates. We didn't do anything for it. But Within that, those who had happened to have eaten more were more likely to lay eggs in the nest. And age, it was negatively correlated. So the younger you are, the more likely you are to start a new nest in the plastic box. In fact, we showed that the probability of new nest foundation decreases with age and increases with feeding. But the most interesting thing was there was no effect on the work being done. The amount of work you have been doing in the parent nest seems to have no influence on the probability with which you will start a new nest. So therefore, we think that current indirect fitness and future indirect fitness are not incompatible. It's not that by working very hard on your parent nest, you forfeit the chances of future direct fitness. And this, I think, is the core result of this. Therefore, we argue that sociality can evolve more easily because workers can gauge both direct and indirect fitness sequentially. It's not that you invest one and you forfeit the other. You can do these two things sequentially. And as you can imagine, this would give much greater opportunities for the evolution of sociality. The third context in which we study new nest foundation is, as I said, putting these artificial nests put together. And these were very interesting because they told us a lot more than just about direct fitness. Very briefly, we set up many plastic boxes, 77 with one wasp per box, we call them singletons, 33 with a pair of wasps, and 30 with triplets, with 3 wasps. Using the same age nest-mate wasps, and observed them until they initiated nests, laid eggs, and produced at least one adult offspring. So we observed all of these, and or of course, or they died, whichever was earlier. And we video recorded the behavior of these singletons, the pairs and triplets. It is the same student who did this project as part of her PhD. And we found some very interesting results. We found the emergence of cooperation and division of labor in this wasp in the interesting way. We found that 2 wasps are adequate for the emergence of reproductive division of labor and cooperation. In other words, whenever we put 2 wasps in a box, if they succeeded in building a nest, they always did it together, never only by one. And one was always a reproductive and the other was a non-reproductive. So both cooperation and division of labor were always found in any successful box in which there were 2 wasps. So 2 wasps are adequate for the emergence of cooperation and division of labor. But 3 wasps are both necessary and sufficient for the additional emergence of non-reproductive division of labor. So when you have 3 wasps, one is the queen, there are 2 workers, but these 2 workers become different from each other. The most fundamental difference between 2 workers is one works at home, one works outside. And when they have this non-reproductive division of labor, then the productivity actually increases. So with 3 wasps, you have cooperation, division of labor, non-reproductive division of labor, and increase in productivity. And we were able to show this statistically because of this very large sample size we have. Between 1 and 2 wasps, there was no difference in productivity, as you see here. But with the 3rd wasp added, right from the beginning, it had an upper hand, statistically significant increase in the productivity. We also were able to use this to study the mechanism by which such division of labor is achieved. Both reproductive division of labor, who lays eggs, who doesn't lay eggs, and non-reproductive division of labor, who works at home, who works outside. As you can imagine, working outside is a more risky task. Working at home is a little bit easier, but of course, laying eggs is the best option. So there are these 3 hierarchical options, and we could study. And we were able to show that both reproductive and non-reproductive division labor were modulated by the dominant subordinate behavior amongst the wasps. In nests with 2 wasps, the egg layer showed significantly more dominance behavior than the non-egg layer, even before nest initiation. And in nests with 3 wasps, the queens showed significantly more dominance behavior than the intranidal workers, those who work at home, which in turn showed significantly more dominance behavior than the extranidal. So the most dominant individual becomes the queen, the next most dominant individual will work at home, and the least dominant individual has no choice but to go and work outside. And therefore, we think there is much more to studying direct fitness options than just refuting a supporting kin selection, than just adding to indirect fitness. So this has now become a paradigm which we find very useful to add to the other ways in which we are studying. Now, as I said earlier, there are two ways in which Ropalidia marginata wasp can get direct fitness. I spoke about one of them. Now I'll speak about the second option, second method of getting it, which I called queen succession. I've already given you and this is a study that we actually been doing a very long time. Even before we actually started thinking of it in terms of direct fitness, because this is a fundamental feature of this society, and we have spent a lot of time trying to understand queen succession. And now we are much more enthusiastically framing this in the framework of direct fitness. Now, I will describe many years of this research very briefly in the form of a series of questions. Research in our group usually begins by watching these wasps. We watch these wasps because we love to watch these wasps. And when you mark them individually with colored spots of paint, they're really a great deal of fun to watch. As we watch them, various questions occur in your mind. Many of those questions seem very difficult to answer, but some of them seem maybe it's possible to answer if I do a little experiment. And then we do an experiment and we seem to answer that question. We have an answer, but we consider that answer only as a tentative answer, because that answer usually raises another troubling question. And until we answer the second question, we are not so sure of the answer to the first question. So we put it aside, we say, maybe this is the answer, and we try to answer the next question. When you answer the next question, you have exactly the same problem. There is a third question that comes up, and this is how it progresses. And I'll give you a flavor of how this happens. In the very first study, I was struck by the fact that queens and workers look identical. Queens can become workers, workers can become queens. There is really no distinction between them. Is in the morphology. Perhaps not much distinction in the physiology. Is there a distinction in the behavior? Do the queens behave in any different way? Can I recognize who is the queen by looking at her behavior? And so the question I asked is, how does the queen behave as compared to workers? Now, in order to answer this question, this is very early. What we did was we marked all the individuals and we measured their behavior. There are two ways in which we measured their behavior. One is we constructed what I call time activity budget for every wasp. For every wasp, we calculated what proportion of its time does it spend in behavior X, in Y, in Z, in all the behaviors, in fact, in 100 behaviors. And therefore, we had time activity budget estimates for every wasp. We also calculated their behavioral profiles for behaviors which are very short duration. Rather than calculate how much time you spend on it, we asked, how often do you do it? How often does a wasp bite another wasp? Per hour, how many times does she bite another wasp? In fact, how often does X bite Y per hour? So with this kind of data, we subjected these data to multivariate statistical analysis, in particular to principal components analysis, followed by cluster analysis. And we showed very early in the game that every colony has three kinds of wasps. These are morphologically identical, so we call them behavioral casts. So we call this process behavioral caste differentiation. And the three groups we call them as sitters, fighters, and foragers based on their major behavior. The sitters seem to spend most of the time sitting, probably apparently doing nothing. The fighters were very aggressive. They were the ones who were showing dominant-subordinate interactions in the colony, and the foragers were really hardworking individuals who spent a lot of time outside the nest and brought back food. Now, deliberately, when we did this, we did not focus any special attention on the queens. We said, if queens and workers look identical, who are we to treat them differently? We put all of this data. The advantage is now, post facto, we could ask, where is the queen? Is she a sitter? Is she a fighter? Or is she a forager? Because we hadn't told our computer who is the queen. And common sense suggested that our queens should be fighters. Because in such primitively eusocial species, which had been studied until then, it was generally known that the queen is an aggressive individual. And she basically rules through physical aggression. She suppresses both worker reproduction and increases worker activity by her physical aggression, by literally physically harassing them. So we expected that the queens would be fighters. And to our great surprise, we found that our queens are not fighters. They are sitters. In colony after colony, queens of Ropalidia marginata are sitters. So the answer to this question is the queen is a non-aggressive, non-interactive, meek and docile sitter. Now, why is this uncomfortable? Because if this were true, there's an uncomfortable question. If the queen is such a meek and docile sitter, how does she become a queen in the first place? Why do the workers respect her? Why don't they challenge her? If the queen is a nasty, aggressive individual, stronger than you, biting you, it's easy to see that you accept. But why should, if there's an individual sitting in a corner there, not interacting, not moving around, why should you accept her as the queen? Why should you refrain from reproduction? And to answer this question, we designed a different experiment. We suspected that if the queen is capable of maintaining reproductive monopoly, and which she does with complete success, she must have some mechanism by which she informs the workers of her presence, of her superiority, and this may be through pheromones. So we decided to ask whether the queen has a pheromone and whether the pheromone is a volatile substance or a non-volatile. So we designed a very different kind of experiment. We studied the wasp, meaning the queen and all the workers on the first day. That is, we constructed time activity budgets, we got behavioral profiles of them. And then on early morning on the second day, with a forceps, we just removed the queen experimentally. The rest of the colony was there, all the workers, and we studied the rest of the colony on the second day, again repeated all the observations, reconstructed time budgets, reconstructed behavioral profiles. And first time when we did this experiment, we were not sure what to do with that queen, which we had removed. We had put her in a little glass vial, we had given her a drop of honey. By the end of the day, she seemed fine. So we said, why not return it? So we returned her to the colony, and she walked in as if nothing had happened and continued to be the new queen. So on the third day, we re-studied the whole colony with the queen return. So we have queen, without queen, and queen return. And these results were extremely surprising because Ropalidia marginata is a fairly peaceful colony. There are fighters, there are a few dominant subordinate interactions, but on the whole, everything is peaceful. But the moment we removed the queen on the second day morning, all hell broke loose. There's a great deal of aggression in the colony. In fact, the aggression on day 2 compared to day 1 can be double, 4 times, 6 times, 10 times. We have even seen a 30-fold increase in aggression on day 2 compared to day 2. This was the first surprise. The second surprise is all of this aggression on day 2 is by one worker, just one worker. And this worker does not receive any aggression. In fact, this is what the data would look like. Day 1, hardly any aggression. Day 2, one individual shoots up her aggression. We go and look at this individual now retrospectively on day 1 because we could not identify her on day 1, and we found that she showed very little aggression on day 1. When we return the queen, interestingly, she drops her aggression. And in fact, if you wait for 2 days, she will drop her aggression all the way. And this is the individual which becomes the next queen. And we call her the potential queen. So our answer is potential queens begin their careers as very aggressive individuals and only later become meek and docile. What was our question? Why do workers accept a meek and docile queen? Answer is the queen is not meek and docile in the beginning. She's very aggressive. That's why they accept her. Now, why is this answer troubling? Because if this were true, how does a meek and docile queen continue to inhibit worker reproduction for the rest of her tenure, which may be several months? She is aggressive only for 1 week. Many individuals are born in the colony after she has stopped being aggressive. Why do they respect her? And in order to answer this question, we designed a different kind of experiment. We designed actually a more complicated experiment. Complicated to do, but very easy to interpret. Now we repeated the previous experiment, day 1 with queen, day 2 without queen, day 3 return queen in a different way. We now closed all the wasps in a cage, closed the doors of the cage, we fed them, didn't allow them to walk. We cut on the first day, we observed the queen and the workers as before. On day 2, we took a blade and cut the nest in half, trying to have equal amount of brood on both sides. We removed all the adult wasps, cut the nest in half, and we introduced a wire mesh screen between the 2 halves. The function of the wire mesh screen was to prevent wasps from walking across, but allow volatile chemicals to go through. After we put in the wire mesh, we had removed all the adults. We took each wasp, we tossed a coin. We said, heads you go left, tails you go right. Next wasp, heads you go left, tails you go right. And finally the queen, people treat the queen like anybody else. We toss the same coin again, heads you go left, tails you go right. And so we had the queen and half of the randomly chosen workers on one side, the remaining workers on the other side. And again, we observed both sides. So one more day, again, time budgets, behavioral profiles for every wasp on both sides of the mesh. On day 3, the equivalent of returning the queen, we simply moved the queen from whichever side she was to the other side. Now, we had very clear-cut predictions of what we expect to happen. If the queen pheromone is volatile, then it should go through the wire mesh. Both fragments should behave like queen-right colonies, and there should be nothing. There should be no difference in the behavior of the wasp between day 1 and day 2, because volatiles are going through and volatiles have the information. However, if the queen pheromone is non-volatile, it does not go through, and the queen-less side should behave like a queen-less colony, meaning there should be a hyper-aggressive potential queen. And we have found this particular experimental design very, very useful. We have used it to answer many questions. I will use it once more in today's seminar, but we use it many times. And therefore, we have done this experiment many, many, many times. When this slide was made, we had done it 24 times. And in all 24 times, prediction 2 was held. The data looked something like this. Day 1, nobody showed aggression. Day 2, one potential queen on the queen-less side. Day 3, that potential queen drops her aggression. A different potential queen on the new queen-less side becomes hyper-aggressive. This is exactly what we had predicted, and that is what happens. So our answer is Ropalidia marginata queens appear to use non-volatile pheromones to inhibit worker reproduction, at least to inform workers of their presence. Now I'll talk about a slightly different kind of question, which arises slightly from this, but also a much deeper question. People always told us that it is interesting. You remove the queen, immediately one big worker becomes hybrid, and only one. Who is this? Can we predict the queen's successor in the presence of the old queen? This, of course, as humans, we are very interested, very curious to know who is the next boss, who is the next one in charge. And surprisingly, we cannot answer this question. We have tried very hard. Many students have worked on this. One student did her PhD on this. She learned many things, but we are unable to predict the identity of successor. Once you remove the queen, within 30 minutes, I can tell you with 100% certainty who is the next queen. In the presence of the queen, I cannot tell you. To give you just an example, one of my students studied many potential queens, many workers, and showed that while the queens are different, the potential queens and workers are indistinguishable. The potential queen is not unique by any criterion that we can measure. Not in her dominance line, not in any behavior, not even by her residual amount of ovarian development. Various workers have low levels of ovarian development. Very different from the queen, but they have some. But in that, she's not special in any way. So the answer really is, we cannot predict the potential queen as long as the old queen is present. Now, for a long time, this was very frustrating to me. But after a while, I got used to it. I said, this wonderful wasp, they still have secrets which I cannot tell. So I'm kind of proud of my wasp that they still have not revealed this secret to us. But students are not as generous as I am. The next student said, we cannot predict, but do the wasps themselves know who the queen successor is? Do they know? Now, for some time, we were dissuaded from pursuing because people said, this is not a scientific question. How can you know the mind of a wasp? How can you know what a wasp knows? But the student was very persistent, and we decided to give a go. We said, let's first frame the question more scientifically, more objectively. And we said, is it possible that there is a hair designate, even though we cannot identify her? And do the other wasps know who this hair designate is? Now, we tried many experimental designs to try and answer this question. Most of them didn't work even on the blackboard. Some of them worked on the blackboard. We did the experiment. We got the result, but we realized that there are alternative interpretations of the data. But finally, I believe we hit upon a design which works. And that involves the mesh experiment. And I illustrate it to you very briefly. The philosophy of doing this experiment was very different. We said, now, for the first time, we are not going to see what happens. We are going to assume that there is a hair designate. All the wasps know who she is. We don't know. We're going to make this assumption. And we're going to do an experiment, and we're going to come up with predictions that should be upheld if our assumptions are correct. If the predictions are upheld, then our assumptions are probably correct. If the predictions are not upheld, our predictions are probably wrong. Now, what are those predictions? We said, look, we are going to do this experiment. We're going to bring the wasps to the cage, put them in a cage, have a wire mesh, queen and half the workers on one side, queen with no queen and half the workers on the side. Now, what is our assumption? There is a hair designate. Now, let us imagine that by chance alone, this hair designate, unknown to us, is sitting on the opposite side of the queen. This can happen by chance alone. Now, what would you expect? This side, there is no queen. and the volatiles, there are no volatiles, non-volatiles don't go through, and we already know that there will be a potential queen. On this side, the hair designate will immediately start behaving like an aggressive potential queen. Let us call her PQ1. Now, if she is the true hair designate, she is not just the legitimate hair for this half, but for the whole colony. Even the wasp on the other side should accept her. So we will put her to the test. We will interchange the places. We'll put the queen on this side and PQ1 here, and this potential queen 1 should be acceptable by these individuals. They should not be attacked. Put her back. There should be no problem. Then she is the true hair designate for the entire colony. However, this will only happen in about half the experiments. Just by chance alone, in the other half of the experiments, the potential queen, unfortunate one, potential queen is sitting along with the queen and she can do nothing. What we expect to happen on the other side, the best among these will become a potential queen. But now she is only the best here. She is not the best for the whole colony. And we test that by again moving the queen and PQ1. And now there should be a problem. The hair designate should be the real hair designate, and this PQ1 should not be. She should stop being aggressive, and the second individual should become a potential queen. But that's not the end of the story. The acid test for this potential queen is now she should be acceptable to the wasp on the other side. So now we interchange PQ2 and Q, and there should be no problem. And this is what exactly happens. So our predictions are the potential queen 1, the first one, should be unacceptable to the opposite side in about half the experiment, and a new one should emerge. But the new one should be acceptable to both sides. There should never be a PQ3. There should never be a third contender to the throne if you are cutting the nest in 2 parts. And these experiments are very tedious to do, although they are easy to interpret. But my student, since this was her project, she persisted and managed to do this experiment 8 times. In 3 of those experiments, the first one was successful on both sides. This corresponds to a situation where she was the hair designate. But in 5, she failed. A new one came, but she was successful on both sides. The potential queen 2 emerged in about half the experiments. No PQ3 emerged, and therefore we think that both our predictions are upheld. And indeed, we argue that there is a hair designate, but we call her cryptic because we don't know who she is. Now, this was a moment of great excitement for us, because every time people ask me, do you know who the PQ is? And I would say no. Now we could say, we know that the wasps know. We in fact published a paper with the title, we know that the wasps know. Cryptic successors to the queen in Ropalidia. Now our confidence that they know comes from a very interesting observation. It does not come from the statistics. It is remarkable that the PQ2 was never challenged. She did not receive a single act of aggression by PQ1 or by any of the workers, either when she emerged in the presence of PQ1 or when she was moved to the opposite side. Therefore, we argue that the hair designate was obviously known and acceptable to all the wasps, including the PQ1. She knew that it's not her turn yet. So there is a hair designate, even though we cannot identify her in the presence of the queen, and the wasp seem to know who she is. The next student came, how do you know that there's only one potential hair designate? Maybe there are a series of hair designates waiting for their turn. Is there a long reproductive queue? I said, we can find out. The experiment was straightforward. What the student did was he took a colony, identified the queen, removed the queen. Then he got a potential queen, identified her. You can do so in 30 minutes. Don't wait for her to lay eggs. Remove her, see what happens. And when he did that, lo and behold, initially no aggression in the presence of the queen. Remove the queen, one and only one individual becomes hyper-aggressive. Remove her, of the remaining one and only one becomes hyper-aggressive. Remove her, of the remaining one and only one, and so on. You can keep on doing this. He did up to 5 potential queens. So indeed, and these individuals were never challenged. I blow up this here. The blue bars, which are essentially 0, are the frequencies with which they received aggression, which was not there. They showed aggression. So everybody knows whose turn it is to show hyper-aggression. There is a long reproductive queue of at least 5 potential queens, but perhaps it is even longer. Perhaps maybe all individuals are in the queue. Therefore, we think that direct fitness, you can see the elaborate arrangement that the wasps have made in order to make direct fitness possible. Not only leaving the nest, having these pre-aggregation conferences, deciding who should be there, as also in terms of queen succession. So there's an elaborate behavioral mechanism by which the provision for getting direct fitness. So we believe that direct fitness may be more important than we have given it credit for, and may even replace indirect fitness in particular cases. So we've really become interested in this concept, and we want to continue to work on this. I want to end by pointing out that you may have noticed that all our experiments were very simple experiments. They didn't cost anything. They did not require any sophisticated laboratory. I'm a great fan for low-cost research. In fact, as was mentioned in the introduction, I've just written a book called Experiments in Animal Behavior, Cutting-Edge Research at Trifling Cost. And here there are 15 examples of experiments of this kind, and there are 16 general praise of low-cost research. This book is freely available on the internet. It's open access. It's available at the link here. You can download it, you can print it, you can do whatever you like with it. And I want to advertise the fact that it is completely free. I also want to say again, as was mentioned in the introduction, I think we should take science to a much broader audience. And as was mentioned, I like to spread the spirit and joy of science. And so I write this bi-weekly column, spreading this joy, and write mostly about animal behavior, ecology, evolution. These are also free, available at the link here, so you can access these. I want to close by saying that, yes, I did not need sophisticated laboratories. I did not need a lot of money, but I needed a lot of brains. And I had a lot of brains to help me. These are all my students who, we would sit. First passion is we would watch the Wasp, then we would sit, we would argue, we would talk, we would do the experiment. So these are my sophisticated instruments with which I've been able to do this work. I'm glad to have had the opportunity to present some of this to you. I will stop here and be happy to take questions. Thank you.
Wow, thank you for a fantastic talk. Wow, it was like listening to a story. It was really a lot of fun. So we have time now for discussion. Before we start the discussion off, I want to remind people of the sort of the format. So please, if you have a question, please type a question mark in the chat. And what I will do is I will relay the question to Rag. And we encourage students and other junior scientists to ask questions. This is a really open group, and I think it's an opportunity for you to ask questions without judgment. And those of you on YouTube, we have someone monitoring that. They will relay questions to me. So there's a bit of a delay, so please just be patient. I'll get you in there. And since we're trying to form sort of a community here, please make sure that when you ask your question that you identify yourself, say your name and where you're coming from. We're all trying to get to know each other better and make this, obviously, as I said, a good community of scientists. So we'll start the questions off. The first one that I have comes from Eduardo, please.
Yes. I just couldn't hold my finger from typing the question mark. We make a big effort of encouraging and letting our students and our younger scholars. But thank you so much. I mean, it's been a fantastic presentation. We couldn't have asked for a better, more engaging presentation that you've offered to us, Rag. Fascinating. Thank you so much. And I just want to, let me see, I wrote so many comments and questions, but I'm going to limit myself to one because this is what we tell that we should be doing. Can you comment a little bit? I mean, you have a lot of data on, you have a lot of behavioral data, and you did get to the pheromones, which it was important to me because I was trying to picture, I mean, are these queens being just passive aggressors? And we can kind of interpret the pheromones as being a passive aggression. What are can you comment a bit on the natural history of their aggressive behaviors? What do wasps do when they're being openly, actively nasty to each other?
So they do 10 different things. And in most of these, it's pretty obvious who is the dominant and who is the subordinate. So a typical thing would be one wasp climbs on top of another wasp and tries to bite her mouth part. And the one which is below crouches and tries not to allow her mandibles to be accessible. So she tries to bite. So we call this one as the dominant, the subordinate. In fact, we call this attack and being attacked. Then wasps chase each other and the other wasp literally runs away. So we have chase and being chased. We have sometimes even more interesting. A wasp, which we call dominant, would come and hold the wing or leg or antenna of another wasp in her mouth and immobilize her. And the subordinate wasp won't be able to do anything. As soon as she releases, she runs away. So there is hold in mouth. They simply sit on top of each other. There is aggression, there is bite, there is peck, there is chase, and sometimes they actually engage in a fight and fall to the ground and have falling fight. So there are many different things they do. We sum up all of these and we compute a frequency per hour of dominance behavior, a frequency per hour of subordinate behavior, and we add up all of these things. That's how we measure.
Any fatal aggression? Do sometimes kill each other?
If you bring a wasp from another colony, they will kill each other. But from within the same colony, we have never seen them kill each other, not even sting. Well, if you bring a wasp from another colony, that's when you usually see these falling fights where they try to sting and they can even kill each other. Within the same colony, we rarely see falling fights, except one very interesting situation. When I didn't have time to talk to you today, but people often ask me, you have this mesh and you have the queen here and the PQ here, and you put the PQ here and the queen here, and PQ goes back to her, right? Now, when will she challenge the queen saying, I am the queen here? So we did that experiment. We wait up to a time. We do this experiment every day. One day, the two will fight. The old queen and the new queen will fight. And that's when you will see a falling fight, where they might actually. So they now have become like two different colonies. One colony has been transformed into two colonies.
Thank you so much.
Okay, our next question comes from Kaya Tombach.
Hi, I'm a postdoc at Hunter College. Thank you so much for that amazing talk.
Thank
I
you.
have a question about group size, because I was struck by the fact that you said that productivity doesn't actually increase with 2 and you have to have 3. Do you see a lot of instability in the groups of 2? And also at the other end, since there's such an extreme, strict hierarchy, it seems, do you see instability at a certain group size when it gets too large?
Yes. We see a few nests which are a single wasp, and we sometimes see them actually go through the entire process of raising at least the first brood to adulthood. We rarely see 2 or 3 wasp nests go through the whole process. So there is a lot of instability. 10, 20, 30, 40 seems to work very well. But once they go beyond that, again, they break up quite quickly. So if I see a wasp with a nest with 100 wasps, I would certainly write a postcard home. That would be very rare. 1 is quite common, but these very small ones, in fact, are quite unstable. You're right. And it's also very convenient for us. 20, 30 wasps is wonderful for the kind of experiments we do. You know, every wasp is marked, everybody has a name, we know everybody what they do. So if it's beyond 100, we need different methods, different techniques to study.
Thank you.
Sure.
Okay, so our next question is from Sean O'Donnell.
Ah, Sean, hi.
Hi.
Mhm.
Fantastic talk. Thank you.
Thank you,
So
Sean.
I'm really fascinated by this process of offspring colonies forming and kind of traffic between parent colonies and offspring colonies. I have a sense, I don't have great data like you do, that this kind of phenomenon is fairly common in tropical wasps. So I'm
Yes.
just curious if you have any sense that there's any kind of pheromonal marking of nest sites or any other kind of chemical communication. I guess one of the reasons I'm interested in this is thinking about the possibility that the lines between independent founding and swarm founding are actually a little bit blurrier.
Yes,
Yeah.
that's absolutely right. It's really blurred here. I won't be able to swear whether this is an independent founding or a swarm founding. It's really, I've had this discussion with Bob Jean for a long time. It is really in between. But you see, in the wild, we never really were able to study this phenomenon because unlike Sirius Sumner, we didn't have radio tracking. But in these large cages, we are able to do this. And what we find is that so wasps leave their parent nest and they have these pre-foundation aggregations where a lot of interesting things happen. And some of them change their mind and go back. Some of them go on to join the new nest. But after going there, some of them change their mind and come back. And even more interestingly, some of those have gone back, later on they change their mind and come back here. So there is this traffic between the old and the new, which we are now able to see. In the field, you only find that wasps are missing, but they come back after a few days. We didn't know what they were doing. Maybe they were lost, and but now we can actually see this traffic going back. And I'm hoping that we will actually be able to study who changes her mind and when and why. So it's now possible to do that kind of thing. But we don't know yet anything about pheromone marking. And we do know, however, that a potential queen on day 0, that is, or day 1 as a queen, has a pheromone profile which is indistinguishable from a worker profile. 7 days later, she has a new pheromone profile which is indistinguishable from a queen pheromone profile. So in over 7 days, she transforms her cocktail. It's a cocktail of some 30 molecules. And the only difference between queen and worker is the relative proportions of the different compounds. But that cocktail clearly gets transformed into a queen type, but it takes a week. And new nests are formed even before that. So it's very interesting, your question. But at the moment, we don't know the answer. Good to see you, Sean.
Great. So our next question comes from Zulema.
Yeah, Sulema Peng Martinez, retired from the University of Missouri in St. Louis. Phenomenal talk, Gadagkar, just loved it. The question I have is whether when you get this new nest founding and you have a new queen, or even in queen succession, presumably the new queens are producing both male and female offspring? And if so, when are they mating?
Okay. Very interesting question. I have to give you the answer based on a lot of things, because we don't have the direct answer. What we find is that in a normal colony, if you dissect all the wasps and look at their spermatheca, we find that some workers are mated and some are not mated, and there seems to be no logic behind it. So it's randomly. What seems to happen, mating never happens on the nest. When females go out foraging, males, of course, leave the nest of their birth and lead a nomadic life. So they're hanging around places where females go for foraging, and they pounce on these females and they mate them. And it appears that the males have no way of knowing whether this female with whom he is mating will ever go on to become a queen or not. But he just mates because he has no choice. Now, if he is lucky, she will go on to become a queen and produce offspring. Otherwise, she will not. So some workers are mated, some are not mated. But we find that when a new colony is founded, at that moment, the queen may not be mated. We have even found in nature, occasionally colonies, where at present the sole egg layer is unmated, producing only haploid eggs. But then the queen will exceptionally go out, get mated, come back, and then produce female offspring. So we have shown by many different experiments that mating itself is not essential for developing ovaries, for laying fertile eggs, at least haploid eggs, for becoming the sole egg layer of the colony, for winning in a competition against a mated individual also it's not needed. So if two individuals, one mated, it's the unmated who may win in the competition, become the queen and mate later.
Fascinating, thank you.
Yeah.
So we have just a reminder to the folks watching YouTube, if you have a question, please send that through to us. We'll ask the question posted here. And others, please keep sending your question marks in. We have the next question comes from Adriana.
Hi, Adriana Maldonado Chaparro from Universidad de Rosario. Thanks for that's a really great talk. Thank you very much. I'm not sure if I didn't get it completely, but my question is, every time you change the potential queen to the new middle of the nest, do they always go through aggressive behavior the first days and then it just goes out? And then have you seen in any of these instances in which aggression goes up or the rate of aggression increases, have you ever seen that a nest is split into by any chance?
The nest never sort of evenly splits into two, but we have data to show that when these small new nests are produced, those individuals who are queens there are actually particularly aggressive individuals in the old nest. So aggressive individuals are likely to leave and start new nests. But except in one study in another species, I have not found a vertical split. But to answer your first question, every time the potential queen begins being hyper-aggressive. And we have often wondered what is the function of this hyper-aggression. Because she's only hyper-aggressive for a week. It's not to control her workers. And we have some very intriguing results. We have evidence to show that the function of the hyper-aggression of the potential queen is not to suppress her nestmates, not to suppress other wasps, but she needs it for her own rapid ovarian development. We think this is equivalent to exercise physiology. By being aggressive, which is a physically intense activity, she's actually able to crank up her ovaries much faster. Now, if this were true, we make a very counterintuitive prediction. If there are only two wasps, one queen and one worker, the queen should be able to lay her eggs very quickly because she doesn't have to waste time and energy suppressing the other one, right? Whereas if there are many wasps, then the queen has to spend a lot of energy time suppressing them. So she should develop her ovaries slowly. We find exactly the opposite. If I am a queen with just one wasp, I have no one to show aggression, I actually take a long time to develop my ovaries because I'm not getting that exercise. Whereas if I'm suppressing many individuals, so queens of multiple foundress colonies will lay eggs sooner than queens of two wasp colonies. So we think that we actually have written a paper saying a possible novel function of aggression. So this is, and in fact, I made the argument that we must focus more on the physiology of the aggressor. We have always been worried about the victim, which is a nice thing to do, but we must also look at what is happening to the aggressor, to her physiology, to his physiology, and what that means. So there is an asymmetry in our attention. I think we should look at aggressors also much more because we have this lead.
Very interesting. Thank you.
Thank you.
So next I had a question that was directed to me, and it comes from Gokul Prabhu. Gokul, do you want to ask it directly here?
Yeah, of course. Hello, I'm Gokul. I'm an undergrad from the Indian Institute of Science Education Research, Thiruvananthapuram. And Professor Gadagkar, it was an amazing talk, and it's always a delight listening to you. So I was just wondering, with the queen removal experiment where you used a mesh setup, it was sort of shown that there is no volatile pheromone signals involved in determining the presence of queens. But
Yes.
was it ever observed if they use some sort of a visual signals to confirm the presence of queen?
We cannot rule out the presence of visual signals by this experimental design. But clearly the non-volatile signals are important. Whether they're augmented by visual signals, we don't know. We've sort of been thinking about this. We can rule out the possibility of unique markings on the face of wasps as Tibets and others find. We don't have any of those markings. And we know that these wasps can sometimes live in completely dark cavities. So we don't have any evidence, but visual signals is not sort of the top of our hypothesis to test, also very difficult to test, but we certainly cannot rule it out. There are also suggestions that there may be vibrational signals. We can't rule that out. We did 1 or 2 experiments to see whether vibrational signals matter, and to the best of our knowledge, it does not matter. But I can't say that we have rigorous proof against vibration or vision. I can't say that. I can only say we don't have evidence in favor of those.
All right. Yes, sir. Thank you.
Thank you.
All right, so our next question comes from Filippo.
Yeah, hi, and this is Filippo Aureli from Universidad Veracruzana in Mexico. And thanks, Raghav, fantastic talk. I really enjoyed it. My question is, a lot of the behavior you describe looks like that the single wasp can recognize the other wasp. I wonder if this individual potential recognition is with all the various wasps in the colony, or there is an obvious recognition because some become either the queen or the possible next queen and so on. So in that respect, there is an individual recognition, or there is a kind of a overall colony recognition, especially in the colonies they are little larger, and then maybe tells something about if there is a dominance hierarchy or there is only dominance subordinate interactions that lead to a dominant individual, not necessarily a dominance hierarchy.
It's very hard to say this rigorously. Of course, there is a colony identity because all non-nestmates are immediately recognized and repelled. So nestmate versus non-nestmate is very clear. Now, within the colony, certainly the queen is recognized. Now, apparently, the potential queen is known from our experiments. Now, for the others, it's very difficult to prove. We have many anecdotal evidences to suggest that individuals know each other. In the way they distribute food, in the way they, in fact, in one experiment, we were able to show that wasps pair together, they would go out and together they would come back. So there are kind of what some people call cliques or subgroups, but whether every individual recognizes every other individual is something very hard to prove. So, but we have anecdotal evidence that must be some individual recognition. It's not just dominance hierarchy, but it's very, very hard to prove. But one interesting experiment which we did was, we see what we do is something I did not explain. But when you bring a foreign queen, she is particularly aggressively attacked. But if you take out the pheromone of the foreign queen, then they don't attack. They, in fact, the potential queen stops being aggressive. So the pheromone, whatever we have said, the Dufour's gland, which we have studied, has information about fertility, but does not have information about foreignness. That must come from something else. So a foreign queen is recognized both because she's fertile and she's foreign, not just one of them. So there is some recognition, but it's going to be very hard to prove that everybody knows everybody by name. This is going to be very hard to prove. I wouldn't know how to do it.
Thank you. Thank you very much.
So we have another question in the queue. Before I go to that question, I want to encourage other students in particular, please put question marks in here. With plenty of time for more discussion. The next question comes from Clara Jones.
Hi, I want to know, can you hear me?
Yes, I can.
Oh, okay. I want to know whether or not you consider the 3 behavioral types to be alternative reproductive behaviors.
We wanted to answer this question, and the experiment we did was in a number of colonies, we classified them, and then we removed the queen and asked, where does the potential queen come from? Which of these 3 groups? And we found that in half the cases, she came from the fighter group. In half the cases, she came from the sitter group, and very rarely she came from the forager group. So we cannot treat them as alternative reproductive strategies. It's more like a division of labor. But in terms of direct fitness options, at least the sitter and the fighter caste seem to be alternative strategies. I can't say that the foraging is a third alternative strategy to get direct fitness, because that seems to reduce your probability of getting direct fitness. So for 2 of them, yes. For the third one, probably not.
Thank you. It was a great talk.
Thank you.
All right, we have another question from Eduardo.
Thank you. You really provided a very, very instructive background to how we think about the primitive eusocial instance and the not so primitive. And I was wondering, are there opportunities? So you have a primitive eusocial insect that can decide whether to become a queen or not after they become the adult stage. Do you have in the phylogeny of these wasps, do you see transitions between what you describe as primitive and non-primitive? And are there opportunities there, if not for you, for one of your younger colleagues, for the next 10, 20, 30 years to engage in a series of questions and more questions and answers to understand what does it take environmentally? What could be the triggers that could get wasps dropping some of the direct benefits in favor of exclusively inclusive fitness benefits?
I love these questions, which allow me to talk about things that I removed because of lack of time. So people always ask me, you spent 40 years studying one species. When are you going to study another species? And my answer is no. I have barely understood this species, and I don't need to study another species. But with one exception. There is a sister species of Ropalidia marginata called Ropalidia cyathiformis, which nests everywhere and often comes and nests in my office. So I couldn't resist, and I began to look at that. And it turns out that these two are ideal to do a comparative study. Ropalidia marginata, where we have done most of our work, is a somewhat unusual primitively eusocial wasp. It is, we have argued, somewhere between primitively eusocial and highly eusocial. It has H polythism, it has a pheromone producing queen, it is somewhat more developed. The other species seems to be a textbook example of a primitively eusocial species. There, the sitters are fighters. And the potential queen is the next most aggressive individual. The queen is an aggressive individual. So that's a typical textbook example, more like a primitive eusocial. This is in between. In fact, right now, one of my students who did a whole thesis on the second species, we are writing a paper arguing that Ropalidia cyathiformis is very primitive, marginata is in between, and honeybees are at the other extreme. So there is this transition. So these two together will give us a very nice way of doing this. And there, in fact, we can show that this is intermediate. And even in our own species, we can have other evidence to say that this is one step above the primitive eusocial. So the caste differentiation is not entirely post-imaginal. So many years ago, I did a massive experiment to ask, at birth, is there some bias? Who will become an egg layer, who will become an egg, or is it completely random? And it is not completely random. I read something like 500 individual wasps, individual plastic boxes. And at birth, I asked, will she be able to lay eggs or will she not be able to lay eggs? And I found that there is a pre-imaginal caste bias. And I found two variables, two things that will predict. Wasps, which eat more as larvae, feel more hungry as adults, eat more and are more likely to become egg layers. Wasps which eat less as larvae, don't feel so hungry as adults, eat less and are more likely to become non-egg layers. So there is a pre-imaginal caste bias. So already in Ropalidia marginata, you are beginning to see the shift from purely post-imaginal caste to pre-imaginal caste. So you're absolutely right, there are these two species. The one problem is that Ropalidia cyathiformis is much more difficult to study. And you'll be surprised why. It is not at all aggressive. The nice thing about Ropalidia marginata is if you mess with it, it will come and sting you, but it will not run away. The cyathiformis, if you mess with it, it runs away. It abandons nest and goes away. And it does not like to be moved to the lab. So it has been difficult to study. 2.5 of my students have been brave enough to do their thesis on this, but 23 others have done on Ropalidia marginata. So there is an asymmetry in our knowledge of the two species, but it would be wonderful to bring that species knowledge to this level, because we really think we have these steps in social evolution by looking at it. And there may be others, of course.
Okay, we have another question from Kaya.
In terms of indirect fitness, do you see any manipulations of which larvae are fed more, any kind of gaming the system by any of the workers or the queen?
That should translate into a higher ability to work, right? Now, there could be, but we haven't looked for anything. So in other words, if you are well fed, well, my impression is that anything that helps you to get direct fitness, and sort of indirect fitness is the sort of default option. Well, of course, I could cheat and say that if you eat less, you get more indirect fitness. If you eat more, you get more direct fitness. The converse of that is if you eat less, you get more indirect fitness because you don't have that. But things that promote hard work rather than ovarian development, never thought about it. I suppose as humans, we would wonder, why would even that be important? But you're right, we should think about it possible because indirect fitness is important. Yeah, no, we've never really grappled with that possibility. But thank you for picking my brain. We do know what makes a worker more fit to be a worker and more fit to leave the nest and start on. That we know, but we have never thought of that as something that is promoting that. We've always thought of that as you know, as a defeat. You can't do that, so you do this. So we haven't thought of this as an active positive thing. But of course we know, in fact, we have built models which will predict what fraction of the population should leave and what fraction of the population should stay. And our field data more or less agrees with those models. So we know what those features are. And one of the important features happens to be the lifespan. If you have a long life, then it is easier for you to get indirect fitness. And we have shown that with models where we are saying that if you have a long life, then you can combine the work of many different workers. So that we know, but we have not thought of it in the way you are framing the question. If there's no other question, I'll take 1 more minute to clarify what I meant by long life. The idea is that if you have a long life, then by yourself, you can lay your own eggs and take care of those eggs, protect those eggs. They become larvae, feed the larvae, they wait till they become pupae, protect the eggs, and wait till the adult comes out who is no longer much dependent on you. But in order to be able to do this, you have to have a very long life. But if you are likely to die before your offspring become adults, then ants will come and eat up everything else. So you will never be able to be a solitary foundress if you're going to die, if you have high chance of dying before your first brood becomes adults. But if you have short life, you can still be a worker in somebody's nest because one worker can work for some time and she dies, but another worker will continue that work. In fact, I have called this assured fitness returns. I've argued that if you want to be a solitary foundress, then you should have long life because you don't have assured fitness returns. Whereas if you are in a colony, workers have assured returns for the investment. I invest for some time and I die. The larva under my care is not going to die. Somebody is going to take care of her. Whereas if I am solitary, then the larva under my care will die. That's why lifespan becomes very important. So one way to answer your question is those who have short lifespan are more likely to get indirect fitness. But that is only because if you had a long life, you would rather get direct fitness. So again, I'm only thinking of it as a loss, as a defeat. But I haven't thought about it in the positive way that you are suggesting I should do.
I guess also I was just thinking as you were explaining that one advantage of a worker that is one of the sort of lower ranked extranidal workers is that it kind of has first dibs on other promising nests if it's going out and foraging and scouting the environment. Do you see those workers be more likely to be solitary founders?
That we don't know for sure, but what we have seen is this. When a nest is small, it's very susceptible to usurpation by other flying wasps. We see that some other wasp may come and join and chase away the queen and take over that colony. So when the wasps are young, their biggest threat is from other wasps of the same species. And in fact, we've even seen when there are several small nests, so each of them has a dominance hierarchy. So here, this one has lost out. She's the worker. So she's not always satisfied with that. She goes out and enters another nest. And sometimes she succeeds in chasing out that queen and becomes the queen there. And this one will go and join the other colony. In fact, there seems to be a period in which there is a lot of flexibility and they are sort of measuring each other. Which is the nest in which I have the greatest chance of being the queen? And I will go choose that nest. So there is some shuffling, quite a bit of movement amongst young nests. It's only when the nests mature or become big, then they don't accept foreigners. But small nests have no choice. You have these usurpers, they drive away the queen and they take over those nests. This we have seen, we have frequented in great, also in quantitative detail, we have documented this.
So I have a follow-up to that, and I apologize if I missed this. When you have potential takeovers happening, who's being aggressive towards the queen, the individual coming in trying to take over?
In these small nests, everybody is aggressive because sometimes you have only 2 or 3 individuals in the colony and everybody is aggressive. In fact, the 2 queens may fight, or the usurper and the old queen may fight, and one of them loses. So in those small colonies, queens also are actually quite aggressive. I showed you in this plastic box experiment that dominant subordinate interactions is the mechanism of reproductive caste differentiation. The most aggressive individual will become the egg layer, the least one will become extra. So at that time, aggression is important. But in the larger colonies, it's not. So the docile queen is in a mature colony.
Thank you.
Sure.
Eduardo, you had a question?
Sure.
Only if somebody who hasn't asked doesn't have one. I don't want to, but
Well, maybe before you ask, let me Kaya, did that satisfy you, to answer your question? Great. Okay, go ahead.
That was great.
Sorry.
Thank you.
it's not as much a comment, a question as a comment from something that we discussed when we met earlier today. We did talk a little bit about natural history and the beauty and the importance of looking at behavior. And now that I've listened to your talk, I realize how much more. I mean, we only talked for half hour in the morning, but through your talk, there was a permanent acknowledgement of uncertainty, of limitations in your studies. I mean, I really made a note. I'm going to use your presentation for the course that I'm teaching on scientific thinking and reasoning, because even when you had lots of stats, again and again, you brought up the issue of, okay, we ask a question, yeah, we kind of have an answer, but still not conclusive. And I think it's very healthy. I would like you to expand on that, because for my taste, we see too much of things being presented and published as case closed. So we figured it out, and we never figured it out. So I'm hoping you can tell us a little bit more how you have now 25 PhDs who have probably listened to you do all this. So can you comment some more on how you approach this? Because I find it very, very motivating, very healthy in how we do science.
There is one trick by which you can motivate people to not to have too much confidence in what they have proved. This is called carrot and stick approach. The carrot approach is to say that the flip side of not having too much confidence in what you have proved is that you can have a little bit of confidence with what you have not proved. If your P is less than 0.05, you can't be so sure. But if your P is 0.07, it may still be right. You may have to do another experiment. So it works both ways. Your success is not guaranteed, but your failure, you're not doomed. If you have that idea, then you can begin to see that there is more to it. The moment P is 0.55, then you're lost. 0.055, you're lost. 0.50, you're okay. It doesn't work like that. So you must have modesty in success, and you must have a little confidence in failure. That's the way I motivate my students.
Quick comment, but then we shouldn't be using the word failure.
Yeah, exactly.
It is right. It isn't
exactly.
failure. It isn't failure. That's what we should be telling them. It is what it is.
There is a continuum, and you are in the borderline between this continuum. And anyway, everybody knows that 0.5, 0.1, 0.05 is all arbitrary. So if you have interest in your hypothesis, which has not been proved statistically, you must continue to do more experiments. You can't abandon. Ideas are not ruled by statistics. Your current data tells you whether you have enough evidence already to support the hypothesis. That doesn't mean the hypothesis is wrong. It simply tells today you don't have enough evidence, but tomorrow you may have. So we have to give more importance to the ideas and use the statistics as a guiding principle.
Thanks. I really wanted you to come because I think that it's important for and this is what we're discussing in the morning, that we will have to put our actions where we put our words. And if we encourage our younger scholars to really follow ideas and look at data and even maybe try to report things that do not seem conclusive, that those of us now at a stage in our lives where we're making decisions regarding you get the grant, you don't get it, you publish, you don't publish, we are going to have to put our actions where we're putting our preaching and be more welcoming of studies that are solid in design, that are moving the arrow a little bit forward without kind of giving us these kind of non-true conclusive statements.
There's a follow-up to that. So of course my students say, okay, my P is 0.07. Can I still say that it may be right? My answer is no. You cannot say the right, but you should believe in the possibility that you can show it to be right tomorrow. It doesn't entitle you. So I, in fact, criticize all paper with saying this is most likely correct because P is 0.06. No, if it is, you decide your alpha. If your alpha is 0.05, if it is greater than 0.5, not today. But you don't have to lose interest in your hypothesis. See, that is where publication is not the end of the world. Your interest, your idea, your future thing is what matters. Publishing is something that is ready to be sent out to the world. What you send out to the world is a small fraction of what you have here. And what you have here is important. So you should pursue that. You know, it doesn't give you, entitle you to publish, but it entitles you to be interested in that person.
That leads me to, there's no other questions in the queue, so just go a little more informal here. You know, we were talking this morning about sharing this with the general public and your blog, and I was just wondering, you know, this ties into this because I think people have images of what scientists do, and you're presenting a very a different view than that I agree with, that than some of us get early on. Could you just speak to how you, I'm not talking about any one particular blog you've written, but how do you speak about the blog in the sense of what you're trying to communicate to the general public and what motivates you to do that?
The first answer is in the name of the blog. It's more fun than fun. In fact, my entire career, my work on Wasp is a hobby. It began as a hobby, and after 10 years of being a hobby, I converted it into my full-time profession. So in fact, I've written an autobiographical piece which says science as hobby. And if you make your science your hobby, then you have a lot of fun. And in order to do that, you should also give yourself freedom about what you want to pursue. If you're too much worried about what the world thinks and what nature will accept or not accept, then you cannot have fun. You should have fun, and so this is what I want to convey to the public, that science is a lot of fun, not just to do, but also to understand. And I try to write my blog in such a way that I make my audience partners in the knowledge production, rather than just passive consumers. And that is why usually I read a paper and become interested. I said, okay, this can become a blog. But that paper occupies 10 to 20% of my blog. The rest is history, the rest is future, the rest is implications. I try to radiate for all different directions, and I always start with what my audience already knows. There should be no gap between the first thing I say and what you know. The first thing I share should be a repetition of what you already know. Then I can add more layers. So whether it's teaching students in a class or giving a seminar to this audience or writing my blog, I try my best to start with something that is already known. And then you immediately get confidence. And also the reader gets, oh, of course I know this, so maybe I can understand the rest. So immediately if there's a gap between audience and you, and then you start, then that gap never gets bridged. And I'm afraid scientists like to do that. There's a friend of mine who used to really make fun of scientists. He says, scientists have the fear of being understood. What's so great? I can understand, so I'm not great anymore. That we have to give up the fear of being understood. Then I think we'll be able to communicate with them.
That's a great point. I'm laughing at it because I don't understand myself sometimes. Any other questions? Well, what I'm going to do here is I'm going to stop the live stream, and then we can have some informal discussion. But I just wanted to say thank you so much for that really fantastic talk and discussion. It's really, quite honestly, this has been like hearing a story. And then you go along with your blog, and not to be cliches, but it really has been fun hearing you talk about your work. And I think we're getting that from the messages I'm seeing and the questions you're getting. So again, thank you for a wonderful talk and for